We just finished talking about this, that DNA has got two big jobs. One job is called replication. That's when you start off with an incredible amount of DNA, a whole cell's worth of DNA, and you duplicate it, and then the cell divides. That's one thing, okay? And it's during DNA replication that we need to understand the binding rules of complementarity, DNA to DNA. But DNA has got a second job, and their second job is transcription. Transcription is when the nucleus takes a little tiny bit of the DNA, opens it up, just so it can make kind of like a little Xerox copy of just one gene. You've got thousands of genes, and when transcription happens, it's not all of them, it's just one. And then that is the way the nucleus tells the rest of the body what it should do. So DNA versus RNA. DNA stays inside of the nucleus, RNA leaves the nucleus. DNA is designed to be permanent. Is it completely permanent? No. But if someone died and their body was buried in the desert 50 years ago, you can still get DNA out of it. As a matter of fact, we have got DNA from woolly mammoths that died tens of thousands of years ago when they were going extinct. And since they landed in frozen stuff, we can get DNA from them. So we're working towards being able to clone a woolly mammoth. Wouldn't that be cool? Anyway, so DNA stays in the nucleus, RNA leaves the nucleus. DNA is designed to be permanent, RNA is designed to be temporary. DNA is divided up into genes, and each gene is the instructions for a protein. This guy, the ribosome, he needs those instructions. The nucleus is not going to lend him the DNA for an hour. The nucleus says, hey, Bob, just follow that little instructions. And then he's supposed to dispose of the instructions when he's done. So the ribosomes are not in the nucleus. The second job that DNA needs to do is transcription, transcription is the term for making messenger RNA, basically. Okay. So DNA and RNA. DNA is double-stranded, RNA is single-stranded, DNA is permanent, RNA is temporary, DNA's got deoxyribose, RNA's got ribose, DNA stays in the nucleus, RNA leaves, DNA's got GATC, RNA's got GAUC. All right, so we left off here. Let's talk about making proteins. We've already talked about the first step in making proteins, which is transcription. Transcription was the step that happens in the nucleus when messenger RNA gets made, and then the nucleus sends that messenger RNA out so that a ribosome will make this guy the little ribosome. He's got, okay, well, I guess this is what I'm supposed to do. When he builds that protein, that is called translation. One of the things you need to do is figure out your way of remembering transcription and translation, because they're different steps. Transcription, basically making messenger RNA. Translation, basically building the protein using the messenger RNA. Okay? Transcription happens in the nucleus. Translation happens outside of the nucleus where the ribosome is. Two steps in building a protein. So figure out your way of doing transcription and translation. Okay? Translation essentially is building a protein. Who builds proteins? Ribosomes, right? Ribosomes build the protein either just somewhere in the cytoplasm, or sometimes they're attached to the rough endoplasmic reticulum. Now, you need 3 different types of RNA to do translation. You need ribosomal RNA. Remember that little R right there? That means ribosomal. The ribosome is made out of ribosomal RNA and protein. Okay? You need messenger RNA. Messenger RNA, that was what Sorry. That's what this little, the nucleus is sending out to, still not going. There we go. Is it worth it? No. Okay, that was a messenger RNA. And then there's one other type of RNA that we want you to know. It's called transfer RNA. And transfer RNA plays an important role. But before we talk about that, let me show you what it looks like. Transfer RNAs are smaller than messenger RNA or ribosomal RNA. Transfer RNAs are kind of shaped like a funky three-leaf clover, a little bit. You should notice that they've got structure, they've got a shape, kind of like a protein does. Now, on one end, they will be carrying an amino acid. And on the other end, they have an area known as their anticodon, right? Now, there are 61 different transfer RNAs. There is one transfer RNA, one specific transfer RNA for every codon that represents an amino acid, right? And whatever their anticodon on this end is, that will determine which of the 20 amino acids they carry. So the transfer RNA, their job is to grab onto an amino acid. Remember, amino acids are the monomers that make up proteins. Building a protein is taking these amino acids and putting them together. This guy, his job is to carry the amino acid to the right spot of the messenger RNA so that the ribosome can hook him together with the other messenger RNAs. Okay, so let's use as an example. The messenger RNA is UUA. We know we're dealing with RNA, right? Because we've got U's instead of T's, right? Now, this is an anticodon. The only RNA that has an anticodon is transfer RNA, which is tRNA. That's the only one that has an anticodon. And by the way, tRNAs do not have codons. Codon is the name for a code word that is part of a little coded message. And the transfer RNA doesn't have any of those. But his anticodon would be AAU. Why? Because do you see how these guys match? Remember those binding rules of complementarity? In RNA, A is going to bind to U, and U would bind to A. Oh, by the way, C still binds to G, and G still binds to C. I did 2 lines between the A and the U, but 3 lines between the C and the G, just to remind us that AU or AT's got 2 hydrogen bonds, CG's got 3 hydrogen bonds. All right. So this guy would be carrying a leucine. Always a leucine, because if the messenger RNA has got this codon, then this guy, this transfer RNA, he goes, oh, that codon matches my anticodon. And the system has worked out that this guy is carrying the right amino acid. Okay? Anticodon. So DNA transcription takes one gene of the whole chromosome, makes an RNA complement called messenger RNA. The messenger RNA leaves the nucleus through a little hole called a nuclear pore, and then in translation, that messenger RNA, which now we call a template, it's a series of codons, little 3 nucleotide code words. It gets read by the ribosome. The ribosome can't really read. The ribosome just attaches and it's like, AAU. And then the transfer RNA goes, oh, my anticodon matches UUA. And the ribosome goes, whatever, and moves down another codon and goes, CUG. And then the next transfer RNA goes, oh, I'm GAC, I match. and it's bringing the right amino acid. And one at a time, it is going to build up a protein. Now, on your quiz this week and on your first midterm, you are going to see charts like this one or like this one. I don't know which one, doesn't matter. You will see them, okay? Now, you need to know how to read them. Step number one, this is a messenger RNA codon chart. It is not a DNA chart, right? It's not a tRNA chart either. It's messenger RNA codons, okay? Now, you know it's RNA because it's got U's in it, and you should know it's messenger RNA because the other types of RNA, they don't have codons. Codons are three nucleotide little code words that are part of instructions for building a protein. So DNA has codons, messenger RNA has codons, the other stuff doesn't, okay? So these are all the codons. By the way, if you ever get a chance to go back in time to the 1950s, bring this chapter of the lab manual with you. This chapter of the lab manual will get you probably 4 Nobel Prizes and the love and respect of the entire scientific community. Either that or they will lock you up, assuming you're from the future, and be scared of you. But one of the two will happen. So, okay, that's how much information is in just this chapter of your lab manual, right? So you need to know how to use this. Let's practice with this one. Let me make myself smaller. Can I make myself? There we go. Make myself smaller. Okay. Every codon has got 3 letters, right? So here's a codon. I don't know what I did there. Okay. Here's a codon, TCG. Is that DNA or RNA? That's DNA. Is this a DNA codon chart? No, this is not a DNA codon chart. Okay, so how did you know that was DNA? It's got a T in it. How do you know this codon chart's not DNA? It's got U's, right? So if you are starting with a little bit of DNA, the first thing you have to do is transcribe it. Remember, transcribing it was doing the rules of complementarity to make some messenger RNA. So the complement of T is A, the complement of C is G, and the complement of G is C. So this will be the messenger RNA. And luckily, this is a messenger RNA codon chart. So first letter is A. First letter is A. All of these guys, their first letter starts with the letter A. So our answer is somewhere in here, all right? Second letter is G. Let's do G. Okay, G. If I go down for the second letter, okay, you can see the place where these two overlap is right here in this little box. So all 4 of these codons start with the letters A and G. A and G. A and G. A and G. And what is the third letter? The third letter is right here. So AGU, AGC. Okay, AGC. AGC is, serine. Okay? Okay, let's do one more. All right? Now, if I give you DNA, you need to transcribe it. What if I give you messenger RNA? Aha! If I give you messenger RNA, do not flip it again. Don't turn that into AUA. Because what is that? That's a transfer RNA anticodon. Now, could I make a transfer RNA anticodon chart? Sure, be really simple. But if I give you messenger RNA, this is a messenger RNA codon chart. So this will just get you frustrated, okay? So if I write it down. If I give you DNA, you need to transcribe it to RNA. But if I give you RNA, you go straight to the chart. Alrighty, so UAU. So the first letter is U. So our answer is somewhere there. Second letter is A. So our answer now is just one of those 4. Third letter is U. U's over here. So tyrosine is our answer. Okay, let's practice. Here is the DNA for the protein that I discovered. Let's just take a little piece right here. Great. So we're going to take a little piece, and then we've got our codon chart. Great. All right, so we're going to start with TAT. Since that's DNA, we have to transcribe it. So AUA. AUA. So A, see how A comes over here? So this is isoleucine. Isoleucine. Great, that's number one. Great. Now we've got AAA. That's the next one. Oh, we got to transcribe it. UUU. So UUU, phenylalanine. It's PHE. Okay. You don't have to know the different names. You'll just, if I give you a codon chart like this and it's got the abbreviations the way this one does, then the answer will just say Lu, Phi, whatever. All right, I think that's it. Yep, that's it. So I will see you. The next video is about how to do DNA fingerprinting and paternity tests.